It's a tremendous honor to receive the 2023 Japan Prize, and I'm delighted to share our work with you today. This story is one that spans the very small bacteria and algae that are single cells to very large structures of the size and complexity of the human brain. And bridging these scales and levels of complexity is a theme that's been very powerful in advancing the study of neuroscience, of biology, and helping us to understand ourselves more deeply. The story goes back, in some sense, a very long time, billions of years, in fact. The original, very earliest manifestations of the complexity of life and the harvesting of light to create energy to support the existence of life, in some ways can be traced back to the archaeal ocean and before, billions of years ago, where the oceans were filled with single-celled organisms, many of which made green light-activated proton pumps that moved a single ion across the membrane in response to a photon. The chromophore, the molecule that absorbs a photon in these biomolecules, is a retinoid, a relatively simple organic molecule, much like vitamin A, and much simpler than the chlorophylls, which have come to dominate energy creation from light on the land as well as in the ocean today. But this ancient form of harvesting light was very efficient and dominant for a long period of time, and the action spectrum of these ancient green light activated proton pumps was very broad, covering most of what we now know as the visible spectrum. These were not information transmission molecules, these were energy harvesting molecules, and this very broad action spectrum in many ways has turned out to be a challenge for us, and I'll tell you more about why that's the case in the present day. These green light activated proton pumps are a member of a larger family of biomolecules that were originally identified by Dieter Osterheld, who passed away late last year. This is a picture of Dieter and his son around the time that he made this discovery of the Halobacterium salinarum bacteria rhodopsin in 1970. As always is the case in biology, these biomolecules turned out to be part of a much broader superfamily. And my colleague, Peter Hagemann, in 2002 described from another single-celled organism, a green algal plant cell, Chlamydomonas reinhardtii, described his observation of a member of this family that was a channel rather than a pump and allowed hundreds of ions to pass through a pore within the molecule in response to a single photon of light. He described this in the same year. My esteemed co-laureate of the JapanPrize, Gero Miesenbach, described his initial steps toward achieving light-activated control of cellular function. A couple years after that, in 2004, I did an unusual experiment. I took a microbial opsin gene, in fact, one from Chlamydomonas reinhardtii, and inserted it into mammalian neurons. This was bringing a gene across major kingdoms of life in an experiment that was very unlikely to work. I'll tell you more about how it worked and what was needed after that experiment shortly. And we also identified shortly thereafter a red light activated member of this channelrhodopsin family, and I'll tell you why having red light actuation instead of the blue light actuation or the green light actuation of these other members of the superfamily, why that was so important. So this collection of findings from single-celled organisms turned out to be very useful, but this is not even as small in scale as some of the important findings actually turned out to be. We've spent a great deal of time and effort and engaged in a fascinating series of discoveries on the high-resolution structures of these biomolecules. And over the years, we've obtained the initial high-resolution structures of channelrhodopsins corresponding to this blue light activated, blue or red light activated cation conducting channel family. We also made the initial anion conducting channelrhodopsins, Peter Hagemann and myself together, and then we got the structures in my group of these anion conducting channelrhodopsins, both the ones that we had made and those that we later observed had been evolved by nature. And these, like the initial ones that we'd observed, were dimers of two 7-transmembrane proteins brought together into a dimer. As it turned out, each member of the dimer had its own pore and its own retinal embedded within the structure. Later, we identified from this very unusual family, these are the pump-like channelrhodopsins. We identified a paradigmatic member of this family, carmine, and we found that it is actually a trimer of 3 of these 7-transmembrane proteins. This pump-like channelrhodopsin family, we found, has interesting ion selectivity. It allows some selectivity for monovalent cations. The blue light and red light activated channelrhodopsins flux sodium, potassium, magnesium, calcium, and protons. But in the pump-like family, ion selectivity can be achieved beyond the cation level. Monovalence being fluxed by carmine, and in fact even potassium alone, or with high selectivity, by another member of this pump-like family. And we obtained that structure just late last year. Now, this effort, all this investigation and discovery on the high-resolution structures of these natural biomolecules has been very interesting. Here is a zoomed-in view of some of these findings. You can see here is that ancient chromophore, the all-trans retinal, embedded within this 7-transmembrane protein. This green cylinder representation of this transmembrane alpha helices is shown here. The gray spheres are water molecules on either side of the membrane. And here these blue arrows are showing the ion flux pathway through the channel pore. The retinal is embedded within a retinal binding pocket shown by these green residues. And over the years, in addition to our structures, we've engaged in a series of investigations on how these residues contribute to ion channel function, both its kinetics, the ion selectivity, and the gating speed of the protein itself. We've been able to powerfully modify what we call photon spike logic, create very fast responses, fast responding channelrhodopsins that can go at hundreds of times per second in driving action potentials in neurons. We've been able to make red light activated channelrhodopsins that are very potent. We were able to make bistable channelrhodopsins that instead of giving you logic of one event per light pulse, to instead flip cells into and out of excitable states on longer timescales. We even were able to create inhibition, creating a molecule called IC++ that turns what's an excitatory action of the channelrhodopsin into an inhibitory action by changing the ions that flux through the channel pore. And so this is one of the outcomes of this basic science work. Understanding the high-resolution structure of the channelrhodopsins has allowed us to create inhibitory channels and actually improve the action spectra, the colors to which they respond. So in addition to changing the kinetics and creating inhibitory channels, we've actually also been able to narrow that ancient broad action spectrum that I mentioned earlier. This is an example of that, the wild-type form of one channel in bright red, and then the one we modified in dark red. Careful structural considerations of the residues around the retinal have allowed us to create this very significant narrowing of the action spectrum, and this has turned out to be very useful for biological applications, as I'll tell you about. Now, it's not just the creation of new kinds of channels that's been interesting. We've come to more deeply understand, as a result of this high-resolution structure function work, how things like channel selectivity, and particularly potassium channel selectivity, can be achieved. Many people watching this will remember the excitement, as I remember, in the late 80s and early 90s when the identification of the primary structure of potassium channels emerged from the Jan lab, and when Rod McKinnon and his colleagues identified the three-dimensional structure of potassium channels. And this fourfold symmetry and the dehydration and mimicking the hydration shell of potassium ions, revealing that this was how potassium selectivity could be achieved, was quite thrilling. It all depended on the fourfold symmetry, and every potassium channel in nature identified since then has had this fourfold symmetry, except the potassium-conducting channelrhodopsins. And our high-resolution structures have revealed that there's no fourfold symmetry anywhere in these molecules. It's a trimer of 3 7-transmembrane proteins, and each of those lack fourfold symmetry within those proteins. And so this has been very interesting, and we've been able to dive deep into the structure relationships that allow ion selectivity to happen in this totally different paradigm. And we've been able to identify a selectivity triad of residues near the extracellular vestibule that actually helps create this selectivity. And we're able to, by measuring the reversal potential, increase the potassium selectivity or decrease it by modifying residues around this selectivity filter. Again, without the fourfold symmetry of canonical ion channels, a new mechanism created by these ancient microbial opsins. Now, this is basic science. It's very interesting how these proteins work, and thrilling to explore as somebody who's long been trained in and appreciated biochemistry. But I'm also a neuroscientist and a psychiatrist, and we want to understand how brains work, and these tools turn out to be incredibly useful. These proteins turned out to be very useful as tools. And our discovery process leading from these microbes to brains has been a long and exciting process dating back now almost 20 years. The initial experiments, just almost 20 years ago, July 1st, 2004, I put a channelrhodopsin into mammalian neurons, and I used a very simple assay for membrane depolarization. These were cells that were not exposed to light. These are cells that were exposed to light. And the channelrhodopsin coupled to a yellow fluorescent protein is seen in this cell, but no activation of the cell as revealed by nothing present in the nucleus, a marker of membrane depolarization, phosphorylation of a transcription factor. This cell was exposed to light and did show this marker of membrane depolarization. And so this was that initial humble experiment showing that one could use a microbial opsin to activate mammalian neurons with light. And a lot has happened over the years. Peter Hagemann and I have had a very long and fruitful collaboration over the decades. My lab has grown from a very small group. Here, the initial group of students, Fung and Ed and Leslie Meltzer on the time of her first paper, Rog Aron, all of these trainees have gone on to great careers in their own right, and the lab has grown quite a bit. But it's interesting to see how far things have come from that very first experiment to the deep level of understanding we've been able to come to of these natural and beautiful proteins. And why does it matter so much for neuroscience? It's because this allows us to achieve a goal of conferring the light sensitivity and therefore actuation capability onto cells defined by type. And this is a goal that has long been held in neuroscience. The electrical and magnetic interventions, although fast, cannot discriminate different kinds of cell that may be right next to each other but will have totally different roles in the brain. But if one could deliver these microbial genes to a cell of interest or a cell type of interest, one could, in theory, turn on or off the cell using light. And this, coupled with the speed of light and crucially, with the simplicity of microbial opsins, a single component protein that encompasses the light sensitivity and the ion flux all within a single gene, this would be very useful for neuroscience. It wasn't enough to put the gene in. A whole host of other technologies had to be developed. We had to develop fiber optic methods and holographic methods for delivering light. We achieved that over the years, notably beginning in 2007. With this paper from Antoine Adamant-Tadis and Feng Zhang in my lab. And an example of the sort of thing that can happen with this fiber optic-based method is shown in the next slide. Here is work studying the basic and very primal mammalian survival drive of thirst. And in work together with Li Chun-lo and Will Allen, a very talented graduate student in my lab, we identified cells in a part of the hypothalamic region called the MNPO that were active during thirst, and we wanted to ask the question, are they causally involved in thirst-related behavior? So we wanted to turn these cells on or off with light. And so we targeted a channelrhodopsin to these cells, and were able to deliver light to actuate the inputs to these cells. And what we found is a behavioral experiment, as you can see here. What's shown here, each little black tick mark is a mouse taking a lick of water, a thirsty mouse drinking water. And we have different trials of the mouse being given a cue, an olfactory cue shown by this dotted line that water reward may be present very shortly. And the mouse starts licking upon getting this cue, and after a time, after hundreds of trials, it becomes sated, it's no longer thirsty, and stops licking for the cue. Then when we drive in that same mouse, if we then turn on the thirst neurons that we had found were naturally active during thirst, but we didn't know their causal effect, when we activated them optogenetically, this sated mouse immediately displayed thirst-related behaviors, the actively seeking and consuming water, despite the fact that it was fully sated. And this is not just a behavioral response, it was a naturalistic brain-wide activity response. We used neuropixels recording electrodes to record neurons from all across the brain, and we found that in all these brain regions, this beautiful response of neurons across the brain. Each horizontal line is a recording from a single neuron with millisecond resolution. Red means more active, blue means less active. This beautiful symphony of neurons across the brain, thousands, with this exhibiting this thirst-related behavior that's abolished by becoming sated, can be fully restored with optogenetic stimulation of the thirst neurons. Nearly complete brain-wide millisecond resolution, single-cell resolution actuation of the thirst response. And then when we turn off the light, that returns back to the sated state. And this is the sort of experiment one can do with these light delivery devices and with these optogenetic tools, come to a causal understanding of what matters for neural circuits in creating their computations and their outputs. We also used our structure function related tools to take away activity of these cells using that IC++ tool I mentioned earlier. And we found that we could take mice that were thirsty and make them no longer seek water nearly as robustly. So gain and loss of function, turning on and off and delivering thereby an understanding of the causal impact of particular cells on behavior. And here, I always like to tip my hat to Francis Crick, who in 1999, very perceptively, long before, or years before these experiments were done, asked for something like this. He said, what we need to do is to be able to turn the firing of one or more types of neurons on or off in the alert animal in a rapid manner. He even thought the ideal signal would be light, and although this was far-fetched to him, he had no idea how to do it. This was exactly what was needed. And his experience, his positioning as somebody who had thought deeply about genetics and biochemistry for so long, he is somebody who was well steeped in the concepts of gain and loss of function, of constitutively active and dominant negative biomolecules that are relevant to biological system function. And this is what we needed in neuroscience, to bring this concept from biochemistry, which is where my deepest roots lie in science, to address the complexity of systems like the brain. The brain is complex, it's heavily nonlinear, it appears redundant in some ways, it's got great complexity, there are many cells that are involved in each behavior. But this is true of many other systems like the genome, like developing organisms. And the complexity is something that can be effectively addressed with precision targeting of gain and loss of function interventions to well-targeted elements. And as in all of these systems, biological systems, one has to be precise with that targeting. And this is what optogenetics allows you to do, deliver actuation to the cells of interest, but brings much more flexibility of timescale of operation as well. Optogenetics allows one to operate at the millisecond scale all the way out to hours, days, or even longer. What's quite interesting is that Francis Crick didn't even ask for single neuron resolution. He asked for control over types of neurons, which is what I've described so far. However, the ability to control single neurons or ensembles of individually identified single neurons is of great value in neuroscience. And this is something that optogenetics has also brought us, this new, not just this new temporal domain, but the single cell domain. And this is where the red light channelrhodopsins that I mentioned earlier became so important. The ways that we have to read out neural activity, we have optical methods that were developed in laboratories around the world. Most of these tools that are optical are blue light actuated, and these include tools, many developed here in Japan, tools at Janelia in Canada, and these all, the most effective, most specific ones, are actuated by blue light. And so if one wanted to do an all optical experiment, control cells, while also reading out activity, one would need a red light activated channel rhodopsin. And this dovetailed very well with our initial work identifying and engineering those initial red light activated channel rhodopsins over the years. And finally, in 2019, we were able to do the following kind of experiment. What you see here are neurons in orbital frontal cortex of an awake behaving mouse, and the waxing and waning of activity that you see in these cells is our control of individual cells guiding spots of light using optics and turning these cells on or off with spots of light and revealing by their fluorescence elevations of calcium inside the cells. Calcium goes up inside neurons when activity goes up, and this becomes a very precise way of identifying activation of individual cells. And you can see all these cells that we're activating in these rainbow colors, T1 to T10, those are the cells we were controlling with individual spots of light. The other cells, which we were not, are shown in gray on the top, the non-targeted cells. Many of these are right next to the targeted cells, but they're not being stimulated in sync. They're doing other patterns of activity that relate to the activity of the animal, but they're not under our control because we're not targeting them with spots of light. Not only were we able to achieve this single cell resolution control, but we were able to show that this mattered for behavior. We saw that some cells were naturally active in orbitofrontal cortex of mice during feeding behavior. Others were not, but were active during social behavior. And we found by stimulating the cells that were involved in feeding behavior, we were able to show they were causally involved. We could turn up the feeding response by activating just the feeding responsive cells. But activating the social cells did not activate the feeding response. Instead, that suppressed feeding. And so it mattered to be able to identify at single cell resolution the relevant targets for optogenetics. Now you could see by that field of view that we had 20 or 25 available cells to stimulate. Many neural circuit properties are going to need more cells than that to control. And here's where yet another value of all of our high-resolution structural work on channelrhodopsins became clear. And that was discovering even newer types of channelrhodopsins that allowed new classes of function to emerge. Our initial crystal structure work, including dating back to 2012 with many of our colleagues, including Osamu Nareki and Hideaki Kato here in Tokyo, allowed us to identify the pore that allowed ions to flow through the pore. And we knew then later, based on our structure function work and turning that pore from a cation conducting channel to an anion conducting channel, we knew what sort of residues lining the pore would predict excitatory channel function. And that, together with some transcriptomic mining, which led to very rich data sets of thousands of neurons, including a collaboration with Susumu Yoshizawa and his team, allowed us to create a very robust collection of possible opsins to explore for functionality in optogenetics, but to narrow down greatly based on the predicted pore structure for those that would be likely excitatory channelrhodopsins. And that led us to discover this carmine channelrhodopsin that is one of the opsins defining this unusual pump-like channelrhodopsin family. It exhibits very high sensitivity, allowing actuation at much lower irradiance values by orders of magnitude than other even high sensitivity channelrhodopsins, and exhibited a red shift in its action spectrum, though still broad like all of these ancient biomolecules are. But its sensitivity allowed us to control many hundreds of neurons simultaneously or asynchronously as we like, and that's shown here. Here, delivering safely powers of light sufficient to control many hundreds of individually specified cells with high temporal precision across 3 dimensions and wide swaths of the mammalian brain during behavior. We used for this purpose holographic devices called spatial light modulators that allowed us to define and project these patterns of light onto targeted cells. And we could do things like have a mouse and train it to detect vertical or horizontal bars and only lick for water to a vertical bar stimulus. And then we could also identify in visual cortex all the cells that responded naturally to vertical bars and not horizontal bars. And we could then provide optogenetic stimulation to those cells, even to dozens or hundreds of those cells, and provide, from our perspective, what seemed like the internal representation of this vertical bar visual stimulus. And we found that indeed the mouse could perform perfectly well with optogenetic stimulation as it could with visual stimulation. There was no difference in its behavioral response, as if we were playing in effectively a perception into the animal. And it wasn't just the behavior that matched the natural stimulus, it was the internal representation itself that arose from the stimulation that was naturalistic as well. In addition to the cells we're stimulating, we're collecting activity patterns of thousands of cells in visual cortex. And we can study the dynamics of this population, reduce the dimensionality, and you can see, identify principal components of these reduced dimensionality representations, and find that the population state trajectories corresponding to a vertical or horizontal perception of a visual stimulus was very similar, naturalistically related to the optogenetic population trajectories. So the internal representation and the behavior of the animal are naturalistic with optogenetic stimulation, as if we're successfully playing in as close to a perception as is definable at the present time. So these sorts of high-resolution, single-cell resolution work, these lines of experiments have allowed us to come to not just an understanding of perception and action selection, but even of memory. And here I want to tell you about some work carried out and described just this year. Now, there's a phenomenon called behavioral timescale plasticity. This was identified by Jeff McGee and his colleagues, including Katie Bittner, back in 2017. What they found was that in hippocampus, a brain structure involved in memory, there was a phenomenon where if they injected current into a single cell, they found that that cell became more active when the animal was in the same context as when the initial stimulation had happened. So effectively creating a place cell, if you will. And this they dubbed behavioral timescale plasticity. Now this was done with a single electrode, hard to scale up, hard to adapt to more high resolution work, but it has always been an interesting phenomenon, and a fair bit of debate has been stirred up in the field as to the mechanism. How might this be working? The intervention is just a direct depolarization of the target cell. Is the change that's happening between here and here, is there a change in excitability of that targeted cell, something within the cell, or is it a synaptic mechanism, a circuit mechanism? And it hadn't been completely clear. What we were able to do in work led by Linlin Fan, a very talented postdoctoral fellow in my lab, was use all optical methods to achieve an optical version of behavioral timescale plasticity. And the things we can do extend far beyond gain or loss of function of individual cells. We can do that, and we did do that. We also were able to tie electrical activity to the genetic identity of individual cells. We could test the strength of functional connections, test synaptic strength. We could ourselves change synaptic strength, and we could measure and quantify excitability of cells, all optically, all using microbial opsins. And this is relatively speaking, in the relatively short time since that initial experiment in 2004, that we can do these immense complexity experiments with all these different axes of optical intervention is really quite astonishing to see. So the experiment that we did, we set up the virtual reality environment for the animal to remain head fixed, running on a floating ball, but we can present it with a virtual reality environment that it moves through according to how it runs on this floating styrofoam ball. And we can control light with spatial light modulators, as I mentioned earlier. What we're doing, though, we're getting a readout of cellular activity, not with a calcium sensor in this case, but with a voltage sensor. And I'll tell you about how this works, a tool that we call Quasar 6A and developed along with Adam Cohen. Adam Cohen and his team sometime back discovered that you could take microbial opsins and you could make voltage sensors out of them. I mentioned calcium sensors before. The voltage sensing, of course, is even faster and is more closely tied to the natural function of the cells themselves, the calcium being a closely following readout of cellular activity. But voltage is much faster, and Adam found some years after our initial optogenetic work showing actuation that microbial opsins could be actually turned into voltage sensors. I won't dwell on this for the sake of time, but we were able to show together with him an improved version of these voltage sensors, the Quasar 6A I mentioned earlier. And if you focus here, this is the operation of this tool. These are natural action potentials playing out, and here are, these can be recorded electrically, but you can also record very similar resolution action potentials optically using Quasar 6A. And so this is an optical readout, and you can imagine the possibilities for probing and understanding neural circuit function using a tool like this. Now, the Quasar 6A is activated by red-shifted light, and so in this case, we wanted the most blue-shifted channel rhodopsin that we could possibly get to pair with it. And so this is the flip side of what I mentioned earlier. Fortunately, our structure-function work dating back to 2012, together with Hideaki Kato and that team, had revealed some mutations that could be used to further redshift, to further blue shift, and that allowed us to create a very potent channel rhodopsin with this particular mutation, T159G, that allowed very robust blue light activated currents and really none at this wavelength at 640, which is the wavelength we used for activating the voltage sensor, the Quasar 6A. So we put these together, the things we'd engineered both on the voltage sensing side and on the optical controlled side, put in the channel rhodopsin that we'd modified there, and then we had the Quasar 6A there, and put it all together and asked what can we probe about how these memory-related mechanisms, how they're actually operating the brain of a behaving mouse. And so here's the experiment working. This is a virtual reality track. The animal receives visual cues that change as it goes over centimeters through this track. And when the animal passes this particular spot, we deliver an optogenetic stimulus. We can activate one or a few or even 10 individually specified cells. And here the cells are being activated when the animal is in this spot in the virtual track. And then we take away the optogenetic stimulus and just continue using the Quasar 6A, the voltage sensor, to observe activity. This is all done optically. And we can see that whereas these cells were quiet before, they're now active in and around these areas in the track where we had done the optogenetic stimulation. So we have succeeded in creating an optically induced place cell that operates in the same way as the electrically induced behavioral timescale plasticity. And this is very long-lasting through many trials, exhibiting all the properties of this particular kind of plasticity. So then, having achieved this, we could then ask mechanistic questions. How is this happening? Is it a excitability change in the targeted cell? Well, we could test this optically using the combination of optical excitation and optical readout. And what we could do is titrate the amount of stimulation we give cells with the channel rhodopsin and observe the spiking patterns that follow that increasing optogenetic excitation and create effectively input-output curves. This is how classically excitability is measured electrically, but now doing it all optically, and we found no change, red versus black, pre and post plasticity induction, even though there was a potent, massive change in the activity of the cell at this spot, a change in its firing properties. It was not a non-specific excitability change. And these are the exact cells that we knew were plastic. So maybe it was a synaptic mechanism instead. Could we identify this As the mechanism, well, this would be an interesting experiment, but technically very challenging. These particular cells in the CA1 subfield of the hippocampus are driven by inputs, including from hippocampal CA2, CA3 regions, but these are close to each other, and we've got a problem because we're already using multiple visible spectrum channels to do our optical excitation and our readout, and here we'd want to separately optically stimulate the inputs to get a synaptic strength measurement. And it's hard to put all those optics together, but my old hippocampal training came to our rescue here. And this is interesting. This just before the pandemic hit, I had been sketching out with Lin in my office this idea, which is to take advantage of the fact that many of the projections coming in to the CA1 subfield actually come from the contralateral hemisphere, coming through the hippocampal commissure, the fimbria and the fornix, and come through, and so one actually could actuate individual cells perhaps on the other side of the brain and have optics that were non-colliding, as it were. And that's exactly what we ended up doing. So we were able to create a ping of cells and observe the inputs as their effect of those inputs on the contralateral side of the mouse. And here are the responses that we can see. We can see little responses, sometimes leading to spiking in the cells that are receiving a ping from the contralateral hemisphere. And then we could ask, does the efficacy of those synapses change during behavioral timescale plasticity? Stimulating the particular cell to induce the place cell properties as shown here. And here red is post-plasticity induction, black is pre-plasticity induction. And you can see we consistently saw bigger responses as shown in red to a ping of the presynaptic cells, revealing that the synaptic connection appeared to be potentiated. And this was a consistent finding, not only at that level, but also in terms of the evoked firing rates that resulted. So it does seem as though there's a synaptic plasticity change happening here, bringing all these different optogenetic tools together to probe different aspects of circuit function. A final question was, what happens if you silence the presynaptic cells? Is the process of plasticity induction, is it presynaptic activity dependent? And there we were able to do yet another optogenetic intervention, come in and inhibit with a pump-like potassium conducting channelrhodopsin that I mentioned earlier, and see if we could block the plasticity induction induced by the postsynaptic depolarization in CA1. And this actually, this beautifully complex experiment actually turned out to be possible thanks to the talents of Lin Fan and the team in the lab. If inhibition of presynaptic activity was achieved by the potassium conducting channelrhodopsin, there was no subsequent creation of place cell properties in that cell. But in the very same cell, if we then took away the presynaptic inhibition and simply did the postsynaptic depolarization, we did create a place cell property in terms of firing rate properties at that spot in the virtual maze. So we were able to come to an understanding that presynaptic activity is required, postsynaptic depolarization, activity dependence, and synaptic changes. All these complexities of this mechanism of plasticity are optical and involve the introduction of microbial opsins into neurons. And so circling back to Francis Crick's initial charge, we can see that what derives from bringing microbial opsins to metazoan biology goes far beyond just gain and loss of function. We can certainly do that, and that's not a small thing. That's the foundation of genetics. That's the foundation of many aspects of biochemistry. That's very important, but we can do so much more. With the voltage sensors, we can link electrical activity with genetic identity of individual cells by targeting those genes. We can test synaptic strength. We can change synaptic strength. We can measure excitability. And again, not that gain and loss of function at cellular resolution is not substantial. In fact, I like to show this slide. This is the famous antennapedia mutation where mice grow legs where the antenna should be. This is a classic pattern formation finding that revealed the logic of body plan formation, but achieved with gain and loss of function intervention in well-targeted genes. And so this is, of course, of enormous significance, and that's gain and loss of function, but we can do so much more than that with optogenetics. In the last few minutes, I want to take a step back from the excitement of what comes to us through the exploration of biology and touch on a little bit of aspects that come from my psychiatry training and opportunities that optogenetics gives us for understanding how things go wrong as well. This is a painting that evokes to me the isolation of depression, the fear and the anguish of major depression, and it makes particularly poignant our inability to see inside, to see what's happening under these barriers that are limiting scientifically and medically as well. The patients I see in the clinic are treatment-resistant depression patients. That's my major area of focus. We've long wanted to be able to come to an understanding of what depression really is in a fundamental sense, and if it's comorbid symptoms like anxiety as well. And the promise of the tools I mentioned is that we may indeed be able to come to a mechanistic understanding of how symptoms related to psychiatry arise. I've written about some of my experiences as a psychiatrist and how these relate to optogenetics and neuroscience, and this book recently did come out in Japan as well. We have a series of stories that are present in the book. They all come from patients that I saw in the emergency room at Stanford, and I tell some of the stories and then go from there to the neuroscience and the technological advances that have given us some insight into the processes at play. Anxiety has been a big part of that, something that mice exhibit very powerfully anxiety-related behaviors. They do other things like us too. They do a lot of parenting and do it well. And these behavioral states are very susceptible to exploration in ancient and conserved ancestral structures of the mammalian brain. We've been able to show that the complexities of anxiety, the negative aversive qualities of it, the risk avoidance behaviors, the physiological changes like respiratory changes, these can be optogenetically induced if you target the right cells and suppressed if you target the right cells. We can show how optogenetically master control regions like the bed nucleus of the stria terminalis, part of the extended amygdala, by sending projections to different parts of the brain, the ventral tegmental area, the lateral hypothalamus, the parabrachial nucleus in the brainstem, can go out and recruit different features of anxiety and create the complexity of this behavioral state. Optogenetics allows us to target cells defined by origin and trajectory across the brain. We and many others have carried out this kind of work. I like showing this work from Catherine Dulac and her colleagues at Harvard studying parenting. Similar story, hypothalamic related nucleus with different projections, one to the periaqueductal gray, another to the ventral tegmental area. She found that the projection to the periaqueductal gray was very important for grooming behaviors in mouse parents, but didn't really affect at all the willingness of a parent mouse to go and retrieve young that were outside the nest and to cross a barrier to go get them. Instead, a different projection from the MPOA to the VTA controlled that behavior, going to retrieve the pups, but didn't affect the grooming response. The parents watching this know very well how grooming and retrieving the young is very important for us as well as for mice, but it can be deconstructed into these two different pathways by resolving individual populations of cells defined by origin and target across the brain. In the last few slides, I want to extend this concept of a brain-wide understanding of the origins of anxiety and anxiety-related behaviors to a body-spanning perspective. And this was something that, again, the pump-like channelrhodopsins that we discovered and developed have allowed us to understand more deeply. Now, body spanning, what does that mean? Well, in the writings of dating back, a very long time, nearly 150 years, William James and his colleagues considered that the fundamental nature of an emotion, although initiated by the brain, because the brain is needed to perceive and make sense of the context that gives rise to the need for and the meaning for an emotion. William James noted that the bodily manifestations of the anxiety are what matter most to us in terms of the quality of the experience. What would an emotion of fear be if you took away all those bodily responses? But this was really an untestable question, because to really test this, you would have to create those bodily responses first, selectively and specifically, and see if those gave rise to the internal state or behavior that was being sought to be understood. And this was not possible to create in a freely moving animal these precisely evoked changes. But a graduate student and a postdoc in my lab, Brian is now a neurosurgery resident at Mass General Hospital in Boston. Richie Chen is just starting now his assistant professorship at UCSF. Did an experiment delivering this very sensitive pump-like channelrhodopsin carmine to the heart of freely behaving mice. And this was important that it be carmine. This allowed us to do non-invasive, freely moving control of the heart rate, pacing the heart using a LED vest in a freely moving animal. And the high sensitivity of carmine meant that we could work at powers that did not cause temperature changes, but still we could precisely set the heart rate of mice anywhere from the resting heart rate of 600 to strongly stress-related, anxiety-related rates up to 900. And the first thing we found was that the mouse didn't seem to care that this was happening, as long as the context was neutral. Here in a place preference task where we only drove the heart, paced the heart in one side of the chamber, the mice did not evolve any preference for one chamber or the other. This is a very sensitive assay for detecting appetitive or aversive qualities of an internal experience. The mouse did not exhibit any preference for the chamber where its heart was being paced even very quickly at 900 beats per minute. But where there was a risky context, whether an open field maze or an elevated plus maze, where the animal has a choice of open arms or closed arms, exposed areas or protected areas, there the behavioral implicit threat that's present seemed to create a different situation. There, it actually mattered, and the animal spent significantly less time in the open parts of the maze when if and when its heart was being paced at the higher rates. So this is a classic anxiety-related behavior, and it was elicited by pacing the heart in this risky context. Similar things seen in other behavioral measures that I'll tell you about in a moment. Now, what could be the mechanism? And so we did an unbiased screen using another one of the technologies that I had developed in the lab now about 10 years ago called Clarity. This allows us to make the brain transparent and look through the brain without sectioning it to look for in this case, activated neurons, activated in terms of driving the CFOS, immediate early gene promoter. And so we looked all across the brain for brain structures that were responsive to the heart being paced at a higher rate. A lot of structures didn't change at all. Primary auditory and visual cortex didn't change. Cerebellum didn't change, but prefrontal cortex cells showed increased actuation, and cells in the insula showed activation. And we followed this up with neuropixels recordings, microelectrode recordings in insula, revealing indeed elevated spiking of a significant number of cells in response to cardiac pacing. And this makes some sense. The insula is known to be important in interoceptive processing. And then we were able to bring in our structure-guided inhibitory optogenetic tool, IC++, that I've mentioned before, and ask which of these structures mattered for implementing the behavioral change, prefrontal cortex or insula. So we delivered now optogenetics in both the heart and the brain at once now, one or the other, or both, and asking what happens if we inhibit insula or if we inhibit prefrontal cortex, what happens to the cardiac pacing induced response. And what we found, if compared to if there was just a yellow fluorescent protein control in insular cortex, here many cells, what the behavior here is an animal showing unwillingness or delayed willingness to seek a water reward if there's a very low probability of receiving an aversive stimulus in response to that. And so these are mice that are very slow to seek the reward if they know there's even a 10% chance of an aversive stimulus coming. And this this response is very much anxiety related, and this unwillingness to seek the reward was powerfully modified by inhibiting, in this case, IC++ and insular cortex. And so this is revealing a causally important brain structure involved in mediating the pacing-induced response. We did not see that effect in prefrontal cortex. The mice that were hesitant to seek the reward continued to be hesitant to seek the reward, even with inhibition of prefrontal cortex. Not that this is not an important brain structure involved in setting up and guiding behaviors, but perhaps operating on longer timescales than we revealed here. And a similar finding with this aversive stimulus gated behavioral task, and also what I've shown you earlier, the open field and the elevated plus maze responses showed similar patterns. So these sorts of experiments have revealed to us that William James in many ways was on the right track, that these internal bodily responses need to be understood, and one must jointly consider the states of cells outside the brain as well as cells within the brain to achieve an understanding of the internal states that matter so much for us in understanding our natural function and our maladaptive patterns. And the fact that, as you can see again and again in these stories, the different optogenetic tools that we've been able to develop and bring to bear, our ability to do that has depended so fully on our achieving these high-resolution levels of understanding of how these ancient single-celled organism-derived proteins operate. And here I want to thank my colleagues, my collaborators, and all those who worked over the years and decades to make this field thrive and to make this avenue of research possible. Particular tip of the hat to Dieter Osterheld, who passed away, as I mentioned just a few months ago, Peter Hagemann, my close friend and colleague, who we've done so much of this work together, and my esteemed co-laureate, Gero Miesenbach. And I want to thank most of all my students and postdoctoral fellows, an amazing team of human beings who have done so much of this work over the years, and their insights have been tremendous. I've mentioned many of our colleagues as I've spoken. I want to also take a moment to highlight Yoon Kim, who is, as you've seen his name appearing again and again on the high-resolution structural work all across the superfamily of channelrhodopsins. A very talented graduate student in my lab, now postdoc in my lab, and a great many other amazing colleagues and collaborators over the years. So I'll wrap up there with a true delight to share this work with you, and humble congratulations to my co-laureate, and thanks to the JapanPrize Foundation and all my colleagues and friends here in Japan. Thank you.